want to do instead is move on to the topic at hand, which is trying to get to a discussion about amino acids and proteins. But in order to do that in the context of all of biochemistry, first I have to fit proteins and amino acids into a broader context of biological polymers responsible for life. And I would argue that there are really four classes of biological polymers that form the basis of life on Earth. You've probably seen this in some capacity in your high school biology course, and maybe you saw it in 701. But the first group, as you are all well aware, right, of course, are lipids. And these are simply alkanes or alkenes that also have a carboxylic acid group. Okay. So, for example, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 14, 15. This is an example of palmitate. Palminic acid. I'm calling it palmitate because I'm drawing it in the anionic uncharged form, C16H32O2. Okay? That's one class. Now, sometimes these are esterified to a polyalcohol, to glycerol, and when that happens, we call them triglycerides because it's the glyceride part is the glycerol polyalcohol. But that's one class of molecules, okay? Alkanes and alkenes. Second class of molecule are carbohydrates. And carbohydrates are literally hydrates of carbon. They have the general formula CH2ON. Okay? And for the most part, these are polyalcohols that either have a ketone or an aldehyde on them. Okay? And an example of that, for instance, that you are all familiar with is that terrible evil molecule glucose. 4, 5, 6. Now, you will notice how many of these carbons that I'm drawing here are chiral. Color this a certain color. Okay, so this, of course, is simply D-glucose. Is it an aldehyde or a ketone? I can't hear you. It's an aldehyde, right? And you remember, I hope, that aldehydes can react, get a different colored piece of chalk, with alcohols. And when I have an aldehyde and an alcohol, aldehyde plus an alcohol, I end up with what? Okay, and this is called a what? Hemiacetal. Right? This is a hemiacetal. And so, although I've drawn glucose for you this way, you can easily imagine what happens. Which is that this hydroxyl can react with this carbon, and in doing so, give rise to this structure. That's got it. This is called alpha D-glucopyranose. It's one of two forms, depending on where I choose on the stereochemistry at this center here. But this is an example of a cyclic hemiacetal. And as you'll learn, I can make long polymers of these. And again, long polymers of these polyalcohols that are essentially cyclic hemiacetals or cyclic hemiketals. That's the second class of biological molecules from which life is based. The third class of molecules are polynucleotides, which of course, nucleic acids, as you know, which are really nothing other than polyesters. So we've seen alkanes, we've seen polyalcohols, and now I'm telling you that the next group is polyesters. Question? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. You're correct. O minus. Thank you for catching that. Okay. Polyesters, okay, as I said, which consist of polymers of the hemiacetal form of a 5-carbon sugar ribose linked by phosphates. And in this case, what we're talking about is a structure that looks like this. The base. And the 5-carbon sugar ribose has a C1 prime substitution. We will go through this in detail later in the semester. C1 prime substitution with a nitrogenous base. It continues down up here, and it continues down here. Okay? And of course, you can see here's one ester, here's a second ester, and so these are phosphodiesters. Okay? So these are polyesters. And the last group of molecules on which life is based are proteins. And proteins, of course, are nothing more than polyamides. Polyamides of alpha amino acids. Okay? Everybody still with me? So all of life comes from 4 types of polymers. Alkanes that have carboxylic acids, polyalcohols, polyesters, phosphate esters, and polyamides. That's all we need in order to create life. Our understanding of amino acids and proteins really starts with physical chemists in the early 1800s. And these people studied the constituents of living matter. And you have to remember that in the early 1800s, the distinction between elements and compounds was kind of fuzzy, and the concepts of chemical bonding were really in their infancy. I'd remind you that Kekule didn't come up with the cyclic structure of aromatic rings until 1857. But the rage back then, the thing that everybody wanted to do, was elemental analysis, right? You could take whatever stuff you had, and as people were learning about atoms, you could basically put it in a a crucible and heat it till it was ash, and you could measure the residue. How much carbon was there, how much hydrogen was there, how much nitrogen was there, and people did this with everything. Tea, coffee, whatever you could find. You could figure out the composition of what it was. Okay? Now, in 1789, before that, there was a French chemist named Fourcroy, and he had identified that from animal sources you could purify three things, three substances that seemed to be related to each other. Today we would call them proteins. They didn't call them that at the time, right? But what he purified were really three animal sources of material. And he called them albumin, fibrin, and gelatin. Okay? And albumin was just a term. Albumin was stuff you got from egg white. It was stuff you could get out of plasma of blood. Fibrin was the stuff that you got if you let blood clot. Gelatin was the stuff you got if you boiled bones. Right? These were just classes of molecules. Okay? And these were, of course, then great targets for quantitative analysis. And the next big insight came from this guy, Dr. George Muller. So he was a Dutch physician who became a chemist, and he focused on trying to understand what it was that was in albumins, egg albumin, serum albumin, wheat albumin. What was it that was in this stuff? And so he did elemental analysis, and I'm showing you the results of what he got here. And the results were absolutely incredible. It looked like if you took fibrin or egg albumin or serum albumin or wheat albumin, which I'm not showing you, they had the exact composition of carbon and hydrogen and nitrogen and oxygen and phosphorus and maybe a little difference in the sulfur. Okay? But everything else was exactly the same. The elemental composition was exactly the same. Okay? 4 different substances, identical in composition. The conclusions were obvious and inescapable. All of these materials in living things must be made of the same thing. They were all made of one substance, which he called grundstuff. Ground substance. Grundstuff. And of course, as you know, if you know the composition, you can write the chemical formula for what this magic grundstuff was. And the chemical formula for this turned out to be (400) atoms of carbon, 620 molecules of hydrogen, 100 molecules of nitrogen, 120 molecules of oxygen, 1 molecule of phosphorus, and either 1 or 2 molecules of sulfur. Okay? And in fact, he then speculated that what must happen, since this was present in plant material and human material and animal material and egg white, it's obvious what must be happening. Proteins must all come from plants, and they must be assimilated whole. When you eat a plant, that grun stuff in the plant becomes the grun stuff in you. Now we laugh at this, ha, 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 ha. But in fact, if you think through it logically, isn't that the logical conclusion you would come to? How many of the things that we think we understand in today's world are the same as this misunderstanding about grun stuff? But for many years, this was the idea. And this magic thing, right? In fact, what he wrote was, he said, quote, "I hold it as now established that the main mass of animal matter is delivered directly from the plant kingdom, and that fibrin and albumin, except for 1 atom of sulfur, have the same composition. The vegetable albumin was synthesized from grain, and the animal albumin was basically the absorption of the entire protein." Incredible. What are we going to call this magic substance from which we are all composed? It needs a special name because it's what we're made of, right? And so we will give this a special name, a Greek name. We will call this magic stuff proteose. Okay? Proteose. Which we now call protein. So here we are with this magic stuff, protein, that we think is one primordial substance that gets absorbed whole, right, and incorporated into our cells. Now, that idea, by the way, was laid to rest by some very interesting observations that were performed on a most unfortunate person. There was a fur trapper in Canada by the name of Alexis St. Martin. And Alexis St. Martin happened to have the great misfortune of being shot in the abdomen. Most people that get shot in the abdomen in those days died. But Alexis St. Martin didn't die. And in fact, what happened was he had a connection that formed where the inside of his stomach communicated now with his skin. He had a fistula. He had a gastrocutaneous fistula. And he was cared for by a famous army surgeon by the name of William Beaumont. He thought, what an opportunity for science. And so he would collect some of the juice, that he would put a tube into that hole and collect some of the juice that came out of it. Or he would take pieces of meat and tie them to a string and dip them in and wait a little while and pull them back out. And then he would look at the juice that came out. And out of this, he made some seminal observations, mainly that, in fact, it did not seem that proteins were absorbed whole, but it seemed like something happened to them in the gastric juice. And this, of course, was the origin of our understanding of digestion. Now, subsequent studies on protein hydrolysates by lots of chemists showed that, in fact, proteins could be broken down into nitrogen-containing acids, which we now call amino acids, okay? So proteins were built from amino acids. Now, just for fun, I will tell you that the first amino acid that was ever identified wasn't recognized as being part, by the way, wasn't recognized as being part of a protein, but the first amino acid that was ever identified, not yet known as part of AA as amino acid, to be identified, was identified by two French chemists, okay? Louis Vauquelin and Pierre Robiquet, and they isolated it from asparagus juice. And they named this, anybody want to take a guess what they named it? Asparagine, of course. They named it asparagine. Okay? And they made a really interesting observation. How many people here have eaten asparagus? How many of you, after you've eaten asparagus, have noticed that your pee smells kind of funky? Okay? That's oxidation products of asparagine. That's what you're smelling, is the oxidation product of that. And they crystallized this, but it wasn't known at the time to be part of proteins. And in fact, the first amino acid to be identified as parts of proteins, you can see in this table, was actually leucine. Okay? And in 1819, and a year later, glycine. And you can see it took over 100 years. It wasn't until 1936 that the last of the amino acids, natural amino acids, threonine, was identified. So with that as our understanding of amino acids, we can now ask, what's an amino acid? And an amino acid, as you know, right, consists of a central carbon atom, which we will call the alpha carbon, and linked to this are four different groups. An amino group, NH2, a carboxyl group, COOH, a hydrogen, shown here, and an R group, a special group that we're going to have a lot more to say about through the rest of this lecture. This R group is called the side chain. Let's do a titration experiment now. Let's do a titration experiment on some amino acids and see what we see. That at physiological pH, at a pH of 7.4, the amino acid is a dipolar ion. Which is a Zwitter ion. And it consists of a protonated amino group and an unprotonated carboxyl group. Okay? Now, we can focus first on this carboxyl group. This carboxyl group has a pKa of about 2, somewhere around 2. And this means now that if I make a plot here where I plot the percent of protonated carboxyl groups, and I plot that as a function of the pH, 7, 10, 11, 13, 14. $150 at a pH of 2. At a pH of 2, what percentage of the carboxyl groups are going to be protonated? The pKa is 2. If the pH is 2, what percentage will be protonated? 50%, right? And if the pH drops below 2, will they be more protonated or less protonated? Who says they'll be more protonated? Who says they'll be less protonated? Okay, talk to the person next to you. I want to know if the pH is below 2, right, whether I'm going to get more protonation or less protonation. Remember, pH equals the pKa plus the log of the base over the acid. Okay, and if the pKa is 2 and the pH is 1, is it more protonated or less protonated? Keep talking. Argue with the person next to you. Sort it out. Okay. How many people think it's less protonated? How many people think it's more protonated? Right. So the percent protonation is going to look something like this, right? In fact, at pH 1, it's 90% protonated. And at a pH of 3, it's 10% protonated. We did this the last lecture, right? So I'm going to get a curve that's going to look like this for the protonation state of the carboxyl group. Now, what about the amino group? Okay. This amino group has a pKa of somewhere around 9. Okay? So that means if I plot here the percent unprotonated amino group, and here's 100, here's 50, 0. When I'm at a pH of 9, when I'm at the pKa, what percent is unprotonated, or what percent is protonated? pH at the pKa. 50%. Okay? So that means here at pH of 9, 50% of it is protonated, 50% is unprotonated. If the pH is, let's make it easy. If the pH is below the pKa, is this stuff mostly protonated or unprotonated? Protonated. Okay? So the unprotonated fraction goes down, right? In fact, the percentage that's unprotonated at 8 is going to be 10%. If a pH is above the pKa at 10, is this mostly protonated or unprotonated? Unprotonated, 90% unprotonated. So in fact, that means that if I plot the unprotonated state, right, of the amino group, I get this orange curve, okay? So anywhere you would agree, I think, between here and here, most of the carboxyl groups are going to be unprotonated, and most of the amino groups are going to be protonated. So I can draw a curve that looks like this. where this green curve is the dipolar or the Zwitter ionic form. Right? So anywhere from a pH of about 2 to a pH of about 9, most amino acids are going to be sitting here, right, in this range where they are, the carboxyl group is unprotonated and the amino group is protonated. If I do a titration curve with glycine, for example, I'll get a curve that looks like this. I'm plotting the pH here versus the equivalence of sodium hydroxide, and I'll find two pKa values, right? One that corresponds to what? What is this pKa of 2? Carboxyl group. And the pKa around 9 or so? Yeah, that's the amino group. Easy. Nothing to it. From now on, we will always write our amino acids. One more important point I have to say. We will always write our amino acids in this form. NH3 at neutral pH. NH3 plus CHR COO minus. This will be the default form that we will draw free amino acids from. Okay? 2 points I want to make. One has to do with charge, which will be important when we talk about purification, right? And one of them has to do with, at one point has to do with why those values are kind of funny. So suppose I plot the charge of an amino acid as a function of pH. 1, 7, 14. 0, minus 1, plus 1. Okay? So, make this 2. What's the charge at pH 7 for an amino acid? How many people think an amino acid like glycine? Okay? How many people think that at pH 7 it's got a charge, it's positively charged? How many people think it's negatively charged? How many people think it has no charge? Yeah, it's a Zwitter ion, right? It has 1 plus charge and 1 minus charge, and so the net charge at pH 7 is 0, right? Now, as you lower the pH, as I lower and lower and lower the pH, what happens? That amino group is always protonated, but what happens to the carboxyl group? When I get to around 2, what am I doing to the carboxyl group? I'm protonating it, right? So the net charge is going to become positive. In fact, it's going to be exactly 1/2 right at a pK, right at around 2.0 where the pKa is. So I'm going to end up with a curve, right, that looks like this so far, right? So at low pH, I'm preferentially positively charged. Now what happens is as I start to move the pH higher and higher and higher and higher, right? At a pH of 7 or 8 or 9, that carboxyl group's already negatively charged. What happens when I get to the point where I start pulling the proton off the amino group? What charge am I going to have then? I'm pulling the proton off the amino group. Let's look at it right here. So now, at that higher pH, right, the amino group isn't charged. The carboxyl group is still charged, so the net charge is, yeah, minus 1. Somewhere up here, it's got to be minus 1. And where is that going to titrate at? Where is it going to be half charged and half uncharged? 9, right? The pKa. So I'm going to get a curve that looks like this. This is my titration curve based on charge. There's something funny about these amino acids, huh? So remember we said that carboxyl group had a pKa of around what? 2, right? But isn't that funny? Here's acetic acid. That's got a carboxyl group. That's got a pKa of about 4.8. How come this carboxyl group's 2 and that's 4.8? That amino group, we said that had a pKa of around what? 9, right? Funny thing, methylamine, that amino group, has a pKa of around 10.6. How come these values are different than free acetic acid or free methylamine? I'll let you guys think about that. Let's talk a little bit about stereochemistry. Oh, did you guys think I was going to answer that? So why does it have a different pH or pKa? Somebody tell me. Say again? It's not resonance, you're close. It's the environment it's in, right? It's the molecular environment that it's in. If you think for a minute about it, in this case, I've got a protonated amino group here, right? And so I have a way to stabilize the anion because there's an electrostatic interaction. So it makes it less favorable to be protonated. And as a result, that's why this carboxyl group protonates at a much lower state. The same way, the free electrons here are being pulled over by the electron affinity of the carboxyl group. So it's a little harder to protonate this. That's why I have to go to a lower pH to protonate the amino group than I do if I just have methylamine, where the methyl group is, of course, pushing electrons towards the nitrogen. Okay? So it's the local environment that controls the pH. This will become important when we talk about R groups.